Good morning, guys. Today we'll start the set of lectures of immune system. So immunology subject, diseases of immune system. It's yeah, as I said you in beginning. So for ones who late, so I repeat it. So this is one of the toughest topics, and you will discuss it also in microbiology. But in microbiology, you also discuss it only superficially. Repeat of this is never enough because this is one of the sides of modern investigations. Yeah, for example, one of the last a few last Nobel prizes are given for inventions in the immune responses, and for example, cancer treatment so now relies on immune response. So a lot of treatments they are based on stimulation of immune response and using of our own immune system to fight against oncological diseases. But first of all, we have to discuss the normal immune response. First of all, we have to discuss normal immune response, unlike immune responses and later we'll discuss the hypersensitivity reactions when our normal immune starts to work against us. So 1 minute. Okay, so immunity, so it's by definition, normal immune response. It defense against pathogens consists of two types of reactions. It might be innate immunity and also it's and acquired immunity or adaptive immunity. So innate immunity is that immunity which present with us just in beginning, so after the birth, and which doesn't require some specific changes during the life. So that's a immunity, which it's like sterile immunity, so you don't have to interact with this bacteria before, yeah, or virus before to fight against it. So and even if you and this innate immunity is a base, but adaptive immunity is like additional structure which helps our body to adapt our body to some outer stimuli. Okay, immunity refers to protection against infections. The immune system is a collection of cells and molecules that are responsible for defending of the body against the countless pathogens that individuals encounter. And part of this we also already discussed in chapter number 3 in a set of lectures of inflammation. And even some of these patterns we discussed in cell injury and cell death. So some events. So today we will repeat it again. So, but some of these points. So, but you have to know it to understand this. Okay, once again, innate immunity. So, which is natural, native immunity. They're always present, poised to react against infections pathogens, and adaptive immunity. It's normally silent and responds on acquired reasons to the presence of infectious agents by generating potent mechanisms for neutralizing and eliminating the pathogens. So first of all, our body is charged to defend against pathogens. And these pathogens might be like viruses, bacteria, fungi, protozoa, or any whatever. So anything which entered inside our body and can potentially provide danger to our to integrity of our body. So innate immunity is facilitated by this. So very superficial we discuss. First of all, it's epithelial barriers. One of function of epithelia is one of the main functions of epithelial cells, so you discussed in histology, which are present all over the surfaces of bodies. So it doesn't matter, it's outer surface, which is made by skin, yeah, or inner surface, which is made by mucosa and epithelia of mucosa. Yeah, so also also every part of our body is separated from another with the help of epithelia. And this epithelia just restrict the sharing of this bacteria, and that point is the essence of this discussion. And but if epithelial barriers fault, So under the epithelia, we have loose connective tissue, so which is battlefield for immune response, in which we have different cells like phagocytes, there are macrophages and neutrophils, which can enter into this connective tissue via the process of inflammation. Yeah, you remember in the process of inflammation, we discussed the recruitment of leukocytes to the site of inflammation, and also dendritic cells, which are acting like phagocytes but has an antigen presenting function to stimulate the adaptive immunity later. So, but dendritic cells, they are part of innate immunity. So, because of their phagocytic activity. Also, here we can remember complement system. You remember that we have two pathways of complement activation, which doesn't require antigen antibody reaction. So when we don't need any kind of specific interaction of our immunity, adaptive stimulation of our immunity, um, so it activates directly, so with C3 activation, yeah, so with the surface, it's known as alternative pathway and lectin pathway of complement activation. I hope you remember this. So it's a first reaction. And also don't forget about NK cells. On NK cells, we have specialized receptors on surface, which can help in activation of also complement system. For example, lectin pathway. So it's promoted with NK cells first of all. Okay, in details we'll discuss each of this event. So, but now let's talk about adaptive immunity. Adaptive immunity is a little different. So, adaptive immunity is mainly promoted by B lymphocytes and T lymphocytes, so lymphocytes. And might be you remember, so in histology, I don't know, I used to talk about it in histology that B lymphocytes and T lymphocytes are, so if we compare B lymphocytes and T lymphocytes with the other WBCs in the blood, it's like phagocytes are like stupid soldiers, yeah, so they just stupid first line of defense, which are soldiers which just follow the instructions. But B lymphocytes and T lymphocytes are much more smarter cells of immunity. They don't act directly. They act in very specific fashion, and they are like officers. If we compare our immunity as defense mechanism, yeah, with defense with army. So NT and B lymphocytes, they act in much more interesting. And B cells. So let me for yes slate that way. Okay, and for example, B lymphocytes, they're charged to produce antibodies, which is typical adaptive immunity event, and antibodies, effects of antibodies. You remember they are first their action. It's activation of complement system, so which is innate immunity, each part of innate immunity, but it promotes promotes this complement system activation for the substances which we didn't used to think that they're pathogens. So that's why we require antibodies, and B lymphocytes are producing these antibodies. So how they produce these antibodies, it's the point of our discussion today. And same to T lymphocytes. T lymphocytes, we have different subpopulations of T lymphocytes, CD4 positive, CD8 positive, and other CD4 and CD8 negative T lymphocytes. And we will discuss it also today. And their main function is so we have a lot of different functions, but one of them is to gain the signal from antigen presenting cells like dendritic cell and macrophages. So with help of main histocompatibility factor, MHC, and promote the other effect of immunity. So might be T killer cells. We have cells, T lymphocytes, which are killing other cells. We have T cells which are activize another lymphocytes. We have T cells which are helping to B lymphocytes to be active. So you see, T lymphocytes are like subdivision of army, yes, so which with special properties, with special responsibility. Okay, and we will discuss it also today. So that's my main idea. Okay, let's talk about innate immunity. So in innate immunity, we discussed a little bit about this in inflammation topic. So we have to remember these effects. They're like TLR. It's known as toll-like receptors. It's a one of the main receptors which are responsible for digestion of new bacterial products. Toll-like receptors so okay, we'll discuss it in details, but they stimulate the phagocytosis by macrophages, so by phagocytic cells. And these toll-like receptors, when they encounter with bacterial products, they promote the cytoskeleton changes and forming the phagosome. Then we have so forming the endosome, yeah, so with the ingested and parts of microbes are destroyed. So we'll discuss it. And then lectin, we have microbial polysaccharides which are present on surface. They are usually specific for bacterial, and we have specific lectin, so which can digest almost every bacteria. So you remember lectin pathway of complement activation. So it's also promoted by leukocytes. So usually it's their phagocytes, dendritic cells, and it might be even epithelial cells. They can express this lectin receptor. Okay, and also inside the cell we have node-like receptors. Node-like receptors, so which are present inside the cytosol. So because they can detect the bacterial peptidoglycan products of damaged cells, so might be responsible for bacterial diminution of bacterial infection, but also they respond on products of damaged cells. So you remember that in necrosis topic, so in chapter number 2, we discussed that necrosis of cells also promotes the immunity reaction. It also promotes the phagocyte activation, and they are activated predominantly by node-like receptors. And we have specific RIG-like receptors, RIG-like receptors. So they are responsible for detection of viral RNA, which is specific response of our innate immunity to viral RNA possession in cytoplasm. So you remember that one of the here I want to stop a little bit. So we'll discuss the infection topic, but I hope you already know that what is a virus. First of all, virus is a parasite. Yeah, so it's intracellular parasite which inject in our cells their RNA or DNA, and it change the activity of entire cell for serving for production of more viral and RNA and proteins of virus. And our innate immunity also cause the formation of RIG-like receptors, which later cause the formation of inflammasomes, so which is also point of our next discussion. So if you now start to bored, so now just embrace yourself. So just little wake yourself up. So now we'll start to discuss it in details. So, major components of innate immunity are epithelial barriers that block the entry of microbes, phagocytic cells, they are neutrophils and macrophages mainly, dendritic cells, natural killer cells, and other innate lymphoid cells, and several plasma proteins, including the proteins of the complement system. The various cells are discussed later in this chapter. The complement system was discussed in chapter number 3. So that's why I realize that you already remember the complement system. Okay, phagocytes, dendritic cells, and many other cells, such as epithelial cells, express receptors that sense the presence of infection agents and substances released from dead cells. So, substances which are produced by microbes, they have one common name: pumps. Pumps. This pathogen-associated molecular patterns. So remember this abbreviation: pumps. So might be here it's shown. No, here it's not shown. So, but later there will be a lot of abbreviations. So you need to remember. So pathogen-associated molecular patterns, as they're known as pumps. Okay, as they are shared among microbes of the same type, and they are essential for the survival and infectivity of the microbes. So, and microbes cannot evade innate immunity recognition by mutating these molecules. So that's main idea. Substances released from injured and necrotic cells are called damage-associated molecular patterns. So it's dumps. Dumps are happen when our own cells are died due to any reason. So might be even physical or chemical or necrotic cells made by ischemia or other type of injury. So that's they produce dumps. But they act almost similar. So, but usually, so just I'm going too fast. But pathogen-associated molecular patterns, they are working first of all on TLR, toll-like receptors. But damage-associated molecular patterns, they are usually act on node-like receptors. So see, this is difference. So remember this difference. TLR is responsible for bacterial invasion, first of all. Node-like receptors predominantly act on non-bacterial products, products of damaged cells. But also they have some effects of bacterial peptidoglycan activation, so as is shown here. So let's talk about receptors of innate immunity. And it's innate immunity receptor. So you see, it's not charged so to bacteria we never encountered with. So it's charged to every bacteria we have, every bacteria so which possibly can enter in our body. And this innate immunity helps us. So patterns recognition receptors are located in all the cellular compartments where pathogens may be present. Plasma membrane receptors detect extracellular pathogens, endosomal receptors detect ingested microbes, and cytosolic receptors detect microbes in the cytoplasm, and as it's shown here. So endosome, yeah, I didn't explain the endosomal receptor. Yeah, toll-like receptors can enter into endosomal parts. So with the process of phagocytosis, yeah. So and when you remember that endocytotic vesicles, they are made up of cell membrane, but cell membrane is invaginated, and they form the endosome. And this endosome possesses the same receptor which were present on cell membrane. That's why we have endosomal receptors. Extracellular, they are lectin and toll-like receptors, cytosolic node-like receptors and regulator receptors, and endosomal, they are also TLR. TLR. Okay, let's talk about toll-like receptors. So there are 10 toll-like receptors in mammals that recognize a wide range of microbial products. The plasma membrane TLR recognizes bacterial products such as lipopolysaccharides. It is a part of bacterial cell wall. And endosomal TLR recognize viral and bacterial RNA and DNA. So same TLRs, they can recognize viral and bacterial genetic information. Recognition of microbes by these receptors activates transcription factors that stimulate the production of several secreted and membrane proteins, including mediators of inflammation, antiviral cytokines like interferons, and proteins that promote lymphocyte activation and the even more potent adaptive immune responses. So let's talk about node-like receptors. Node-like receptors, as I said, there are cytosolic receptors named after the founding members Node 1 and Node 2. They recognize a wide variety of substances, including products of necrotic cells, might be uric acid. You remember, it's uric acid is a product of degradation of RNA and DNA, and released ATP. So ATP must be kept inside the cells, inside the mitochondria, but if ATP is lose their arrangement, so if it released into the cytoplasm, it causes activation of node-like receptors. So also they are sensitive for ion disturbances, for example, loss of potassium, and also for some microbial products. So only for some microbial products. So node-like receptors are not predominantly charged to microbial products, but predominantly for our own cells. Okay, how this family sensor detects so many diverse signs of danger or damage is not known. So it's a point of investigation now. Several of the NLR signals, not like receptor signals, we are cytosolic multiprotein complex called inflammasome. So inflammasome, we little talked about inflammasome. It's not like receptor protein, so which collect other proteins like adaptors and caspases. So which causes the inflammasome, which activates the caspases, and you remember, so it pro-interleukin 1 beta, so causes the activation of interleukin 1 beta, and this interleukin 1 is promotes the inflammation. So you see, inflammasome is activated by node-like receptors. Node-like receptors causing the formation of inflammasome. Inflammasome activates the caspase 1. So you remember, caspases are cytoplasm proteins which promote the apoptosis. But this caspase 1 causing the formation from interleukin 1 beta, pro-interleukin 1 beta, the active form of interleukin 1 beta. So which we already discussed, it's one of the main cytokine of inflammation. So this interleukin 1, so which is produced as a result of this activation. Okay. So it's activation on node-like receptors. But here you see, here we can mention that TLRs, they toll-like receptors, which are activated by bacteria, they also promote the formation, and other receptors of inflammation. So they activate the pro-interleukin beta gene activation. Yeah, you remember that they act on nucleus and they promote the synthesis of pro-interleukin 1 beta. So you see, activation of only node-like receptors is not enough to promote the inflammation. So you see, it's intercalated system is present here. So why it's present? So to be sure that this immunity response is really important, is really necessary for us. So that's main idea of that's the main idea. Okay, let's talk about other receptors of microbial products. We have C-type lectin receptors. CLRs, later we will discuss them. They are expressed on the plasma membrane of macrophages and dendritic cells. So going furtherly, we can say that these macrophages and dendritic cells, they are antigen-presenting cells, and they require to digest this bacteria in specific pattern, and they detect fungal glycans and elicit inflammatory reactions to fungi. So several cytosolic receptors detect the nucleic acid of viruses that replicate in the cytoplasm of infected cells and stimulate the production of antiviral cytokines. Okay, you also remember we discussed a little bit about G protein coupled receptors on neutrophils. They're also present in macrophages and most other types of leukocytes. They can recognize short bacterial peptides containing this N4-methionine residues. So don't need to remember it by heart, but just remember we have some bacterial, so only bacterial compound which promotes the formation of G protein coupled receptors. Okay, and this we have mannose receptors recognize microbial sugars, which often contain terminal mannose residues, unlike mammalian glycoproteins, and induce phagocytosis of the microbes. This is also activity of lectin receptors. You remember it in lectin pathway of complement activation. We discussed about mannose endings, yeah. Which are interacting with lectin, and due to this, so it causes the activation of cascade of reactions. Okay, we have more families, two families of cytosolic receptors. They are named after the founding member RIG1, and the other called the cytosolic DNA sensors. They recognize microbial RNA and DNA respectively. So RIG recognize RNA, DNA sensors, so as the name implies, recognize DNA. So this diagram is discussed. So loss of potassium from the cell, causing the also formation of inflammasome and activation of NLRP, node-like receptor protein. Okay, reactions of innate immunity. So which reactions of innate immunity we know is there inflammation. So as we discussed before, this promotion of synthesis of interleukin 1, so interleukin 1 beta, so it promotes the inflammation and causing the all these effects, all the events we discussed before. So the causing the interleukin 1, remember, the causes the activation of integrin family formation, integrin, so activation of integrins, they also causing the formation of more selectins on the surface of endothelial cells. It also promotes the activity of leukocytes, so to this site of reaction, and other effects we have. So which are like cascades are going faster and faster. And in chapter number 3 we discuss it in details. And antiviral defense. Of course, antiviral defense is, for example, type 1 interferons. They produce the response to viruses act on infected and uninfected cells and activate enzymes that degrade viral nucleic acids and inhibit viral replication. So that, as we discussed, present here. Yeah, so this Reg-like receptors, they act on it. Reg-like receptors which are present in cytoplasm, they also can destroy viral RNA. And also endosomal receptors, not like same as toll-like receptors, they also can recognize DNA, and their DNA recognized receptors are also present in cytoplasm and cytosol, and that's how they facilitate the antiviral defense. Okay, in addition to these defensive functions, the innate immune system generates signals that simulate the subsequent more powerful adaptive immunity response. And this we'll discuss as adaptive immunity. Okay, so you see, this innate immunity, they promote the activity of adaptive immunity also. And this, let's talk about adaptive immunity. Adaptive immunity system consists of lymphocytes and their products, including antibodies. So, in contrast to the limited repertoire of the innate immunity system, the adaptive immune system can recognize a vast array of foreign substances. So, what's the positive parts of adaptive immunity? What is the negative part? So, adaptive immunity is very targeted to some specific pathogen. So if we have some specific pathogen, so if it's not recognized by our innate immunity, so it can be recognized by this adaptive immunity. It's a good thing, yeah. So because response goes much faster, and in complement system, I explained it to you, yes, that classical pathway goes much more effective than alternative pathway, and also lectin pathway. So it goes much more effective because much faster cascade of complement is activized from C1. But if activation goes from C3, so it goes not so abundantly, and we need to directly split C3 here. But if it cascade starts from C1, it can cause the splitting of more C3 and formation of more C3b on the surface of this bacteria, and that's why this cell will die much faster. And also, okay, and other events. So we have a lot of events. But on this example, I can share you the idea what's the importance of adaptive immunity. Sometimes a lot of our bacterial cells, bacteria or viruses, they start to act like start to mimic to the to our own tissues, and we need to reveal so which are pathogens, which are not pathogens, and that's why we need adaptive immunity to reveal these pathogenic parts. Okay, so and there are two main cells, as I said before, which are responsible for adaptive immunity. There are B lymphocytes, or known B cells, and they provide the antibody production. And T lymphocytes, they facilitate cellular immunity, so cell-mediated immunity, T cells. So as I mentioned before, I don't remember, in some practical classes I said it, but might be not to your group, that difference between T lymphocytes and B lymphocytes is like, so just imagine two assassins, yeah, two assassins. One assassin is killing with a bullet, yeah, with a gun. Another one is killing with a knife. So, and B lymphocytes, they're like guy with a gun. Yeah, so with a gun, so can shot into from the distance into the enemies. But T lymphocytes, they have to come closer to this enemy and kill it directly by activation of their components. They're T lymphocytes, especially T killer cells. We are talking about T killer cells. Okay, antibodies provide protection against extracellular pathogens in the blood, mucosal surfaces, and tissues. So we'll discuss types of antibodies, but you already heard about them. It's there IgE, IgG, IgM, IgD, and IgA. So, and for example, mucosal surfaces are followed with IgA, immunoglobulin A. Okay, and T lymphocytes are important in defense against intracellular microbes. They work by either directly killing infected cells, accomplished by cytotoxic T lymphocytes, as I said before, or by activating phagocytes to kill ingested microbes. For example, by the production of soluble protein mediators called cytokines, and they're produced by helper T cells. So you see, helper T cells don't directly kill this bacteria, but they activize it, so just mark it with cytokines to attract more phagocytes to kill this bacteria. And that's also one of functions. Yeah, so without knowing where's the direction, in which direction to kill, yeah, so our macrophages don't act very effectively, so we need to show where to kill. Okay, cells and tissues of the immune system. Immune system so usually comprises to the lymphoid organs. Yeah, so you heard that the lymphoid organs are responsible for a immune system. And now we just interact the innate and innate immunity, so effects of innate immunity and adaptive immunity. Okay, let's talk about lymphocytes. So lymphocytes are present not only in blood, and they are present in lymphoid organs like diffuse lymphatic tissues. They are present in lymph nodules. Also, they are present in lymph nodes. They are present in spleen. In thymus and even in bone marrow. Yes, we have different lymphoid organs. We have primary lymphoid organs and secondary lymphoid organs. You discuss it in histology. No need to discuss it, I think. And the primary lymphoid organs, they generate the lymphocytes, but storage of these lymphocytes going in secondary lymphoid organs. So primary lymphoid organs are bone marrow and thymus, but usually the cells are final destination of the cells is the lymph nodes, lymph nodules, and diffuse lymphatic tissues. Okay. So each T or B lymphocyte and its progeny, which constitute a clone, expresses single antigen receptors, and the total population of lymphocytes, numbering about 10 in 12 in humans, can recognize tens or hundreds of millions of antigens. So that's my idea. They are directed to some specific antigen. Okay, the enormous diversity of antigen receptors is encoded by variant DNA. So, as you remember, first lecture we discussed that we have DNA. We have DNA, so which is so we used to think that DNA is information, and it's like kin coded cell, yeah, and it directs what to produce. But we have some part of variant DNA, so which are changed. Along with especially it's typical for B and T cells, and they change the combination, and successful combination of this DNA causing the secretion of proper cell membrane cell surface receptors, for example, or even antibodies. Which are produced by B cells. So that's why variant DNA is very important, especially for immunity cells. So they are joining and diversification of different gene segments to form functional antigen receptor genes, a process that occurs only in B cells and T cells. That process happens only there. Hence, the presence of rearranging antigen receptor genes is a reliable marker of the cells. As already mentioned, the antigen receptors that are expressed in B cells are called antibodies, and T cells, they known as T cell receptors. So remember, T cell receptors, they're known as TCR, and everywhere you can find TCR, it's abbreviation TCR, so it's a T cell receptor. I'll remind it to you. Major T and B lymphocytes recirculate through peripheral lymphoid organs. So recirculation means entering into the bloodstream and then enter back into the lymphoid organs. So like lymph nodes, spleen, mucosal tissues, and reside in these organs and in most tissues. So foreign antigens are concentrated in these organs where they bind to and activate the clones of lymphocytes that express receptors for those antigens, a process known as clonal selection. So once again, so we need to produce clones. What is the similarity of clones? Similarity of clones is DNA sequence. But all our cells are similar in sequence DNA, yeah. But because of variant DNA, our immunity cells, they possess different DNA sequences, and that's why we shouldn't produce all these B lymphocytes and T lymphocytes, all of them in large amounts. So we need to produce some specific type, which is responsible for only this bacteria which we encounter with. So that's why we need to select the B lymphocyte and T lymphocyte, which will be reproduced. And when antigen comes to this area, so it gives the stimuli to this B lymphocyte and T lymphocyte to produce the monoclonal population. So with the same genes, with the same rearranged genes, so which are necessary to fight against this bacteria. All mature lymphocytes go through distinct phases during their lives. They might be naive lymphocytes. They express antigen receptors but have not responded to antigens and do not serve any function. So they need to be maturated. Then effector lymphocytes, they are induced by lymphocyte activation and perform the functions that eliminate microbes. And also memory lymphocytes. After every bacteria, so some, especially usually B lymphocytes, they are circulating in our blood for a long of time. So their population is decreasing during the time, but sometimes they're present all over the life, or might be so. But in some cases, they're present in our body for few so 10 to 20 years. And if our body encounters with this infection again, so this memory lymphocytes, they start to activize, and they start to produce the monoclonal population of these cells, and they can cause the response. And that's the position which we require for the process which is known as vaccination. Yeah. So in vaccination, our task of vaccination is produce this memory lymphocytes. And if our cells are already encountered with this antigen, so we have memory cells. And then if even large amount of this bacteria entered in our body, or virus, or fungi, doesn't matter, our memory lymphocytes, they can reproduce themselves, and in very fast way. So they produce enough antibodies and T cell receptors which can kill this offending agent. So that's the main point of memory lymphocytes. Okay, so let's arrange a little break for 2 minutes. So just keep information in your mind. So we'll continue in 2 minutes. Okay, after a little break, so let's discuss T lymphocytes. T lymphocytes are known as T lymphocytes because they're thymus-derived T lymphocytes. They develop into the effector cells of cellular immunity and help B cells to produce antibodies against protein antigens. They constitute approximately 60% to 70% of the lymphocytes in peripheral blood, and are the major lymphocyte population in splenic periarteriolar sheaths. So you remember this pus, yeah, which is present in PAC, which is present in spleen. So we discussed it in histology. And lymph node interfollicular zones. In lymph nodes interfollicular zones, they're present T cells. So you remember that in lymph nodes we have lymph nodules, yeah. So and peripherally they're present with T cells. And in interfollicular zones, in follicles we have lymph nodules, but interfollicularly we have T lymphocytes. T cells cannot recognize free or circulating antigens. Instead, the vast majority, 95% of T cells, sense only peptide fragments of proteins displayed by molecules of the major histocompatibility complex, MHC. So let's explain what is MHC. MHC is present on phagocytes. The MHC is present on dendritic cells and macrophages. MHC is a complex which it's antigen presenting complex. So MHC is like scaffold for presenting of antigen to these T lymphocytes. And if antigen is appeared on MHC, so our T lymphocytes must to act on this, and must to produce, so must to kill this type of antigen all over the body. So this MHC, and MHC, we have MHC1 and MHC2. So what's the difference between them? So actually they work as similar, but they work for different cells. What is MHC1? What is MHC2? So because T cell antigen receptors have evolved to see MHC bound peptides on cell surfaces, T cells only recognize antigens presenting by other cells. So you see, T cells can't recognize bacteria by innate immunity. That's why T cells are only responsible for only adaptive immunity. So T cells working only with, so they act only on MHC-bound peptides of proteins. Okay. Peptide antigens presented by MHC molecules are recognized by the T cell receptors. T cell receptors, as I said before, yeah. So Faizan just joined, yeah. So interesting. T cell receptors. So without biochemistry of these T cell receptors, so okay, let's show it on this diagram. Yeah, T cell receptor is composed of. So we have two different T cell receptors. They are made by CD4 positive and CD4 receptors and CD8 receptors. We have CD4 and CD8, and they are accompanied with other subpopulations like CD3. We have zeta chains, and also sigma chains and epsilon chains. So it's a very deep biochemical molecular biological question we are not going to discuss today. And you see, MHC molecule. So we have two types of MHC. We have class number 1 and class number 2. So in class number 1, class number 2 MHC molecule interact with CD4 positive cells. So okay, main idea. T lymphocytes are mainly they have two populations: CD4 positive, which express this CD4 receptor, and CD8 positive, so which express CD8. Yeah, and lymphocytes, T lymphocytes, which are present with CD4, they interact only with class 2 main histocompatibility complex molecule. CD8 interacts only with class 1 main histocompatibility molecule. That's main idea. And this main histocompatibility complex, which is present on antigen presenting cells, as I said, macrophages and dendritic cells, they just show this peptide antigen. So which was so how it's appeared here we'll discuss a little later. But main idea, when bacteria is digested by macrophages, so some part of their peptide is present on the surface of the cells by main histocompatibility complex, and it matters. So if it's a class 2 main histocompatibility complex, it is interacting with CD4 positive lymphocytes. If it's first, it interacts with CD8. Mainly idea is similar, but it has little differences, so we will discuss a little later. So that's CD4 positive. It interacts with second, CD8 with the first. Okay. It's important in pathogenesis of some diseases. So that's I explained. CD4 is expressed on 50 to 60% of T cells, whereas CD8 is expressed on about 40% of T cells. So a little less CD8 is present. CD4 and CD8 expressing T cells called CD4 positive and CD8 positive cells respectively perform different but overlapping functions. CD4 positive T cells are called helper T cells, so this remember this name. T cells are which express CD4 positive, they are helper T cells. Because their main function is to help other leukocytes to be activized. They secrete soluble molecules that help B cells to produce antibodies and also help macrophages to destroy phagocytosed microbes. So you see, CD4 positive cells, they are not killing cells by themselves. They're just helping to produce antibodies and also activize other macrophages. And central role of CD4 positive helper cells is in immunity is highlighted by the severe compromise that results from destruction of these cells by human immunodeficiency virus. So CD4 positive cells are first of all digested by HIV, so which is described later in next lectures, I mean. But CD8 positive T cells, they also secrete cytokines, and they are known as cytotoxic T lymphocytes, or sometimes called T killers. Cytotoxic T lymphocytes. That's why you can see the abbreviation CTL, cytotoxic T lymphocytes. But on the surface of leukocytes invariant, so in both of them we have CD28, so which functions as to receptor for molecules called co-stimulators. They co-stimulate the activation. So without activation of CD28, so normal immune response doesn't happen here. So and actually our so why we discuss about it. So one of the latest Nobel Prize is given for finding the way to. Okay, you know that our immunity fights not only against bacteria, it finds also against our cancer cells, for example. And these cancer cells are they block the CD28. They somehow block the CD28. It's a very interesting interaction. And the main activity of this new drug which was invented is a prevention of blockage of CD28. It's activation of the CD28 which causes the normal cytotoxic effects on the cancer cells. Okay, we will discuss it later, I guess. T cells that function to suppress immune responses are called regulatory T lymphocytes. So they are not CD4, they are not CD8 positive. They are necessary to facilitate the tolerance of self-antigens. Our body expresses a lot of antigens, so we have a lot of proteins which potentially can cause the inflammation against it. But main idea is that we have mechanism to prevent that activity. And you see that impairment of regulatory T lymphocyte activity so cause the autoimmune reactions. Okay, while the most T cells express T cell receptors composed of alpha and beta chains, a minority of peripheral blood T cells, and many T cells associated with mucosal surfaces, for example, lung, gastrointestinal tract, express T cell receptors that are composed of gamma and sigma chains, which are similar but not identical to alpha and beta chains. So such gamma and sigma T cells, which do not express CD4 or CD8 receptors, recognize non-protein molecules, for example, bacterial lipoglycans, but their functional roles are not well understood. Another small population of T cells expressed markers of both T cells and NK cells. They are known as NKT cells. Recognize microbial glycolipids and may play a role in defense against some infection. May play a role, it's not confirmed. That's why no need to stuck your attention too much on this. But if you become a researcher, so keep your attention on this too. The antigen receptors of gamma sigma T cells and NKT cells recognize antigens independently of main histocompatibility complex. So that's specific role of this. So I said before, yes, CD4 positive and CD8 positive, they can gain the antigen only from antigen presenting cells with the help of MHC. But here it doesn't require MHC molecules, and they are much less diverse than the receptors of the more abundant CD4 positive, CD8 positive T cells. So these populations will be not discussed later because their function is not well understood yet. So might be in later books and later research it will be confirmed. So which role in immunity they play, and must be it's very important immunity part. But we remember one thing, that we need reactions of CD4 positive and CD8 positive reactions. Okay, now let's talk about major histocompatibility complex molecules. Major histocompatibility complex molecules, they are fundamental to T cell recognition of antigens and genetic variations in MHC molecules. So they are molecules are associated with many immunologic diseases, hence it is important to review the structure and function of these molecules. Main histocompatibility complex was discovered on the basis of studies of graft rejection and acceptance, like histo, which is tissue, and compatibility. It is now known that the normal function of MHC molecules is to display peptides for recognition by CD4 positive and CD8 positive T lymphocytes. In each person, T cells recognize only peptide displayed by that person's MHC molecules, which are the only MHC molecules that the T cells normally encounter. This phenomenon is called MHC restriction. So that's the main idea. So T cells cannot be activized without MHC. Human MHC, so sometimes you can find this is like human leukocyte antigen, so it's HLA. So that it might create some confusion for you. So in some books you can find that it's what is HLA. So HLA and MHC is a similar thing. It's absolutely similar. HLA and MHC is similar. Okay, so don't confuse it. And so that's why in some books, in some studies, it will be named as HLA. So because it's to concentrate our attention that it's human, but MHC is present in mammalians, yeah, and other animals. Okay, on the basis of their chemical structure, tissue distribution, and function, MHC gene products fall into two main categories. We have class 1 MHC, which are composed of HLA, HLA-A, HLA-B, and HLA-C. Each of these molecules consists of polymorphic alpha chain on convolutions associated with an invariant beta-2 microglobulin peptide. So no, don't remember this. No, doesn't matter this. The extracellular portion of the alpha chain contains a cleft where the polymorphic residues are located and where foreign peptides bind to MHC molecules for presentation to T cells, and conserved regions that bind CD8, ensuring that only CD8 positive T cells can respond to peptides displayed by class 1 molecules. So you see MHC and MHC1, which is made up of HLA 1, 2, and 3. So they are responsible only for CD8 positive T cells activation. So, and CD8 positive cells, I remind you, they are T killer cells. They are cytotoxic T cells. They cause a direct activation of these T cells. And class 2 MHC, so they are encoded by genes in the HLA-D region. So genes of HLA-D region, they have DP, DQ, and DR. So don't remember. So unlike class 1 MHC molecules, which are expressed on all nucleated cells, expression of class 2 MHC molecules is restricted to a few cell types, mainly APC. So it's antigen-presenting cells. So especially dendritic cells. Also they're present in macrophages and on B cells. So you see a second type of MHC present only in immunity cells. MHC1 might be present by every cell of our body, almost every cell of our body. So that's a main difference. So several other proteins are encoded in the MHC locus, including complement components like C2 and C3 and factor B, and the cytokines tumor necrosis factor and lymphotoxin. So TNF is also present in the same locus. Okay, so let's over this HLA. So then after this, so we'll continue this lecture in next week. HLA genes are highly polymorphic. That is, there are alternative forms of each gene at least at each locus. Their estimated number over 10,000 for all HLA genes and over 3 and 500, 3,500 for HLA-B alleles alone. So each individual expresses only one set of HLA genes. It is believed that the polymorphism of MHC genes evolved to enable display of and response to any conceivable microbial peptide encountered in the environment. So, polymorphism, a virtually infinite number of combinations of HLA molecules exist in the population. And actually, this okay. HLA genes are closely linked to chromosome 6, so they are passed from parent to offspring and block and behave like a single locus with respect to their inheritance patterns. So why do we discuss it? So why we have this discussion? Actually, we require it to understand that we check this HLA in cases of transplantation. So HLA patterns. So we have different types of this HLA. So we usually check for six types of HLA, and if more than five, if five or more HLA proteins might be similar. If they are similar in two patients. So this we can easily transplant this organ without a risk of rejection. And usually it happens only in relatives, so that's why usually we have transplantation from relatives, right? But if we have a HLA which is different, so yeah, we have to live on immunosuppression all the life after the transplantation. So that's why it's very dangerous, and you know that immunosuppression causes the bacterial infections and viral infection. Every viral infection might also even normal usual cold, yeah, can cause a death in these patients. So because we need to depress the immunity state of these patients. Okay, it's also will be discussed later. So this is how genes of HLA are promoted here, so it's not so, yeah, we'll skip it. So it's not so, it's important thing, but not for everyone. So if you need it, so if you're going to work as transplantationist, you of course need to know it. If you want to work with transplantation of organs, you have to understand the HLA genes. But actually, every day this idea of HLA compatibility so is discussed, and every day so found something new. That why when you became a doctor, so if it will be required for you, so you will learn by heart. Okay, B lymphocytes. So we'll start to talk about B lymphocytes with the next lecture. So next lecture is also abundant with explanation.
मोहसिन माइक ऑफ कर
So Maxine in douche now, yeah. So here I think he decided to wash his body, yeah. Okay, now let's check your attendance. So 1 minute. It's recorded, and also check your chat. Okay, thank you. See you next time. So your chat messages are recorded. So once again. So now you can leave. Thank you for attention. See you next time.
ओके थैंक यू सर